Hi, I'm Ramya. In this talk, I hope to convince you that data science approaches applied in tandem with high throughput experimentation can revolutionize the way we design, synthesize, and evaluate polymeric biomaterials. Since my talk will be in the context of gene editing, I'll begin by introducing what gene therapy is. In cells with defective gene sequences, the production of functional proteins is compromised. By introducing genetic material in the form of DNA or RNA, we can augment, delete, or modify these defective gene sequences, restoring healthy cellular function. In the clinic, this is typically done using engineered viral vectors, which have gotten to be highly safe and efficient over the years. A downside to using viruses is that it pushes the price of these gene therapeutics well beyond the reach of ordinary patients. In this graph, we can see the prices of recently approved gene therapeutics. Highlighted in red is Zolgensema, which costs 2 million. This is not the price per month or the price per year. This is the price per treatment infusion. Obviously, this is the only therapeutic option available for a debilitating disease called spinal muscular atrophy. And if we look at the driving forces behind these exorbitant price tags, we can identify three causes. First, around 30% of the product cost arises not from the therapeutic DNA or the RNA, but from the use of viral vectors. Second, the FDA spends around 80% of the review time auditing quality and manufacturing issues associated with viral vectors. Finally, pilot units that produce clinical grade viruses for trials, they are backed up and they have wait lists as long as 2 years. If we could somehow replace viruses with a synthetic material that's scalable, affordable, and readily available off the shelf, gene therapeutics would be significantly cheaper, much easier for the FDA to regulate, and also they would make their way faster through clinical trials from the lab. Now, polymers are considered highly promising alternatives to viral vehicles, but we have some design considerations. Polymers form polyelectrolyte complexes with polyanions in the form of therapeutic nucleic acids such as DNA, RNA, and ribonucleoproteins. Ribonucleoproteins will be the focus of my talk today. The polymers themselves are polycations incorporating positive charge centers either in their polymer backbone or in their pendant units. Now, thanks to advances in controlled radical polymerization, we can make almost any arbitrary combination of architecture, composition, and molecular weight possible. Despite these enormous advances in polymer synthesis, we still don't have too much guidance on how polymer structure and composition affects its ultimate biological performance. And this is compounded by the extremely large design spaces opened up by polymers. For instance, if we look at the simple design framework of a linear copolymer, we can look at repeat units of varying basicity and comonomers of varying degrees of hydrophilicity. We can incorporate these two repeat units in the form of block copolymers or statistical copolymers. We can vary their composition to make them, cationic dominant or hydrophilic dominant, and also play with the degrees of polymerization. If you look at all possible combinations, it gives rise to 256 unique structures. Obviously, it's not feasible to synthesize and test every polymer in this portfolio, and we also have very little guidance from theoretical models that can predict biological performance directly from the structure. In order to solve this problem, I propose using combinatorial designed polymer libraries, high throughput experimentation, and machine learning to accelerate the rate of discovery of polymer vehicles. By applying machine learning techniques to identify structure function relationships governing polymeric gene delivery, we will be maximizing the information value of each and every experiment, not just the polymers that work, but also the polymers that are extremely inefficient. So, how this workflow operates is as follows. First, we will use combinatorial design to synthesize a polymer library, which will be subjected to rigorous characterization using a suite of physicochemical techniques. We will formulate polyplexes with this polymer library and identify hit polymers through various screening assays. We will also evaluate the editing efficiency, cytotoxicity, and the cellular uptake of all the polyplexes in our polymer library. Finally, We will integrate the large data sets we obtain from physicochemical characterization, hit identification, and biological evaluation in order to identify structure function correlations that will give us some experimental guidance for designing the next iteration of polymer libraries, where hopefully we can locate an even greater number of hit polymers. So I will start by describing the rationale for my polymer library. We looked at cationic monomers spanning a range of pKa values, uh, ranging from monomers like AMA composed of primary amines, as well as monomers incorporating tertiary amines with alkyl substituents of varying steric bulk. We also looked at hydrophilic comonomers incorporating zwitterionic groups PEG as well as HEMA. So, for each pair of cationic and hydrophilic monomers, we targeted polymer compositions ranging from 100% cationic to 0% cationic. In this study, we used a parallel synthesizer which can conduct up to 12 raft reactions in a single experiment. But moving forward in the future, I would like to explore continuous flow polymerization methods in tubular reactors to boost the synthetic throughput further. Now, in this combinatorial scheme, we have 43 structures, and I will just be showing you a snapshot of the characterization data obtained from the HEMA subset of copolymers. As you can see, for most part, we were able to achieve our targeted composition, and thanks to the raft kinetics, we were also able to obtain highly uniform distributions of polymers. Finally, we also measured the pK and the zeta potential of all 43 polymers in order to understand their protonation behavior in greater detail. Having synthesized the polymers with the desired molecular weights and compositions, we proceeded to formulate polyplexes with RNPs at different charge ratios and evaluate gene editing. The cell line we used for assessing editing performance provides a very convenient fluorescent readout. Here, the Cas9 protein is guided by the guide RNA to the target region in the genome. It results in a double-stranded break at the target region, which is repaired through endogenous cellular processes. The resulting insertions and deletions can push the mCherry sequence into the reading frame. About one third of these frame shift mutations will result in mCherry production in edited cells. It is important to note that even if you have 100% editing efficiency, only one third of the edited cells will actually express mCherry. This obviously demands a screening assay that unites throughput as well as sensitivity to low editing efficiencies. We turn to image cytometry, wherein we used an automated microscope to acquire multiple fields of view for each treatment condition. This microscope was equipped with a motorized stage, and we were able to collect a large data set of images. This data set was passed through an image processing pipeline that could classify each cell as either mCherry positive or negative completely in an automated fashion. And it was able to compute the fraction of mCherry positive cells for each treatment condition, and the data is summarized here in the form of a heat map. As you can see, the vast majority of our polymer library turned out to be completely ineffective at mediating RNP delivery and editing. However, we had one exception, the 50-50 statistical copolymer of HEMA and the diisopropyl-based cationic monomer, both of which are highlighted in red in the chemical scheme. This particular structure exhibited editing efficiencies comparable to a commercial control. So we took this forward for further evaluation, and we performed flow cytometry to further characterize its editing performance. We benchmarked this polymer DIP50H50 against four state-of-the-art commercial controls, which are highlighted in blue. And we can see that even at an NTP ratio of 1, our hit polymer outperforms all four commercial reagents, including RNP specific reagents such as CRISPR Max and JetCRISPR. Sanger sequencing also mirrored this trend, and we were able to observe that the hit polymer resulted in editing efficiencies as high as 60%. Having identified a promising polymer for further development for in vivo as well as ex vivo editing applications, we turned our attention to a different question. We asked ourselves whether the low RNP delivery performance of the rest of the library originated in low cellular internalization, or was the editing efficiency compromised despite high levels of cellular internalization? In order to answer this question, we employed fluorescently labeled RNP constructs, which allowed us to quantify cellular internalization across the entire library. By doing this, we wanted to identify the mode of failure for all the other polymers that did not perform as well as our hit polymer. So, what we noticed from our internalization studies was that predictably, our hit polymer exhibited high levels of RNP uptake. Most of the library had RNP uptake that was comparable to unpackaged RNPs, with the exception of two polymers. The statistical copolymers of AMA and HEMA exhibited RNP uptake levels comparable to the hit polymer. These two polymers are highlighted with black stars. These near miss polymers did not were not limited by cellular internalization, but they still had extremely low editing efficiencies. So we asked ourselves why these polymers were unable to unpackage their payloads once they bypassed the cell membrane and entered the cells. To answer this question, we turned to confocal microscopy to measure the degree of colocalization between the RNP payloads and lysosomal compartments. What we saw was that the hit polymer was less likely to get entrapped within lysosomes compared to the near miss polymers, which tended to co-localize with the lysosomes. So it appears that for a given polymer to be successful, it needs to satisfy two sets of design rules. The first set of design rules is for guaranteeing entry into the cell. The second set of design rules is having entered the cells, can they actually unpackage the RNPs within the cytosol so that these RNPs can make their way to the nucleus and initiate genome editing. To figure out what these design rules were, obviously we wanted to identify the structure function relationships governing both uptake as well as delivery. So we looked at nine different polymeric attributes which we thought could be implicated in RNP delivery as well as internalization. However, deriving these structure function trends purely through pattern recognition or intuition turned out to be close to impossible. We recognized the need to apply more sophisticated approaches, and we turned to a machine learning technique called random forest classifiers, which is a tree based classification scheme. I won't get into the details of how this algorithm works, but what our models told us was that for cellular toxicity, it is largely a function of polyplex size, with bulkier polyplexes mediating more toxic responses. As for RNP uptake, we found out that electrostatic interactions were critical. If you can engineer a polymer that can have a high degree of protonation at physiological pH, such that its pK and zeta potential are high, then you're more likely to interact with the cell membrane and enter the cell. Finally, I want to emphasize that electrostatic interactions alone are no guarantee for success. In order to actually unpackage the payload and realize genome editing, we also have to pay attention to hydrophobic interactions. We found out that what differentiated our hit polymer from our near miss polymers was that it had a higher value of C log P, which is the measure of hydrophobicity, as well as the Hill coefficient, which is a parameter describing the degree of cooperative deprotonation. So our hit polymer was able to balance the demands of both uptake as well as RNP delivery, whereas the near miss polymers were only able to satisfy the demands for uptake. I'll be talking more about how exactly random forest classifiers were applied to mine these structure function relationships in my talk on Wednesday. I'll also be talking about the Hill coefficient quite a bit on Wednesday. To wrap up this talk, I want to say that complexity is inevitable when synthetic biomaterials interact with the physiological environment. One way to deal with this complexity is by leveraging advances in automation, robotics, and high throughput experimentation to explore diverse chemical spaces and to discover materials from domains in the chemical space that you normally ignore. And the second thing is there does exist a trade off between resolution and throughput, and it can be balanced by doing high resolution studies later on in your discovery phase rather than earlier on. Finally, This discovery of our hit polymer may have been serendipitous, but we don't have to let this happy accident go to waste. We can apply machine learning tools to derive design rules that can be applied for synthesizing future iterations of polymer libraries, where perhaps we can discover even more high performing polymeric vectors. I'd like to thank my advisor and my lab mates for their support. I'd also like to thank the funding agencies and the Imaging Center at the University of Minnesota. I'm grateful to the AIChE session chairs and other organizers and all of you for listening to my talk virtually. I encourage you to get in touch with me